Greetings to all. I'm Eleni Naziri and I'm an assistant professor of food biochemistry of the University of the Aegean in the Department of Food Science and Nutrition on Limnos Island in Greece. My lecture is Microbial Pathways to Sustainable Lipid Production from Food Waste Streams and is provided by the AGRIMA project, which runs under the Erasmus Plus project. The name of the AGRIMA project is Agri-food Waste Management for Sustainable Bioeconomy through Higher Education Curricula and Upskilling. The goals of AGRIMA is to foster university capacity building for the green transition through innovative practices and higher education curricula updating in agri-food waste management for the circular economy. AGRIMA addresses advancing pedagogical methods for industrial agri-food waste valorization based on business-academia synergies. Additionally, addresses integrating citizen science in bioeconomy enhanced waste valorization as a means of civic engagement and environmental advocacy. The consortium consisted by the Polytechnical School in Porto, in Portugal, which is also the coordinator of the project, Aix-Marseille University from France, AVIP Association from Portugal, University of Novi Sad from Serbia, and also, except from the University of the Aegean from Greece, the Web to Learn. The key points that will be covered in this lecture is the definition and the type of lipid compounds. We will also discuss the roles of lipids in food and human health. We will go on with the fatty acid and especially with the essential omega fatty acids. We will discover the sources of lipids and why there's a global need for new lipid sources. And then we will go on with the main part of this lecture, which is the oleaginous microorganisms and their metabolic route and production strategies. We will go on with the industrial bioprocess workflow and also the sectors that these lipids or single cell oils that they are also named, that they found applications. Then we will discover some food waste that they can be used as substrate for these oleaginous microorganisms for lipid productions. And we will go on with the challenges and future directions in terms of technical, economic, and environmental consideration. We will end up with some specific case studies of agro-industrial and food waste used as substrate for lipid production. Generally, lipids are a diverse group of compounds that share a key chemical property. They are soluble in organic solvents, but not in water. This hydrophobic nature defines their role and behavior in biological systems. Within this broad category, we recognize several major types. As you can see in these slides, we have the fatty acids, which are the building blocks, the triacylglycerols, which is the main form of energy storage. We have also waxes, phospholipids, which are essential for membrane structure, sterols, which play both structural and signaling roles, and also we have the fat-soluble vitamins and pigments which support various physiological processes. Lipids provide 9 kilocal per gram and they are used by the body for long-term energy storage, particularly in adipose tissue. They also act as carriers for fat-soluble vitamins like A, D, E, and K, without which sufficient dietary lipid absorption of these vitamins is compromised, leading to potential deficiency and associated health issues. Moreover, lipids contribute to the sensory properties of food, serving as sources of aromatic compounds that are able to enhance flavor, especially during baking or frying. Moreover, they are a structural precursor for steroid hormones such as testosterone and estrogen, and phospholipids form the basic matrix of cell membranes. Additionally to their metabolic and structural function, lipids are crucial in determining also texture and mouthfeel of foods, influencing creaminess, smoothness, and flavor release. Certain lipids are able to act as emulsifiers, stabilizing a mixture of oil and water, as seen in lecithin in mayonnaise. In the human body, subcutaneous fat serves as an insulator layer, reducing heat loss and helping maintain a stable body temperature. These combined properties explain why lipids are vital not only for health, but also for the sensory and physical qualities of food that we consume. In dietary planning, the reference range for macronutrients, as you can see in this slide, ensures that energy needs are met while maintaining optimal health. Lipids are typically recommended to provide a specific proportion of total daily energy intake, 15 to 30%, which is enough to support essential physiological function without contributing to excessive caloric intake. This balance is key because both deficiency and excess of dietary fat may lead to adverse health outcomes. Now, let's go on to the structure. Fatty acids are the simplest form of lipids consisting of a hydrocarbon chain with a terminal carboxylic acid group. This structure creates two distinct ends, as you can see in the slide. A polar head and a hydrophobic non-polar tail formed by the hydrocarbon chain. The length of this chain and the presence or absence of double bonds largely determine the fatty acid's physical properties, such as the melting point and the solubility, as well as the biological functions. Now, the fatty acids can be further divided in the saturated fatty acids, the saturated fatty acids, such as the palmitic acid seen in this slide, contain no double bonds in the hydrocarbon chain, meaning that each carbon atom is fully saturated with hydrogen atoms. This straight chain structure allows them to pack closer together, resulting in a higher melting point and making them solid at room temperature. In terms of nutrition and health, saturated fats are have specific metabolic effects, and their intake is often monitored for cardiovascular diseases. The unsaturated fatty acid can be divided into monounsaturated and unsaturated fatty acids. The monounsaturated fatty acids contain a single double bond within their carbon chain. This creates a slight bend in the molecule, preventing tight packing and lowering the melting point, so they are often liquid at room temperature. A key example is oleic acid, which is abundant in olive oil, and this consumption is linked to beneficial effects on cardiovascular health. Monounsaturated fats are regularly considered as a healthier dietary lipid option compared to higher level of saturated ones. The polyunsaturated fatty acids play a vital role in human health, contributing to the development of the nervous system, offering cardiovascular protection, preventing inflammatory disorders, helping to reduce serum triglyceride levels, and preventing effects against cancer. The unsaturated fatty acids have multiple double bonds contributing to flexibility and influence how the fatty acid interacts in biological membranes. As you can see in this slide, I give you some examples of linoleic and linolenic acids, with have 2 and 3 double bonds respectively in their structure. The position now and the number of these double bonds determine their classification and their function in human metabolism. The polyunsaturated fatty acids are considered as essential fatty acid and are divided as omega-3, omega-6, and these are polyunsaturated fats essential in the human diet. Their name, using this omega system, comes from the methyl end of the carbon chain. For instance, omega-3 means that the first double bond is 3 carbons from the end. Because humans cannot synthesize them, we must obtain them through diet. Some of the most well-known omega fatty acids is linoleic acid, classified as omega-6 because its first double bond is 6 carbon from the omega end, and alpha linoleic acid an omega-3. Oleic acid is an omega-9 essential fatty acid, while arachidonic acid, another omega-6, plays a role in inflammation and brain function. The diversity in this example shows how small changes in molecular structure can translate into very different biological functions. A balanced dietary ratio between omega-3 and omega-6 is key for supporting brain health, cognitive performance, and overall mental well-being. Now that we have seen which fatty acids are essential, it's important to ask where do we actually get these lipids from our daily diet. So lipids come from both animal and plant sources. Animal-based products include milk and dairy, as well as meat and meat bioproducts. From the plant sources, we have olives and, of course, olive oil, soya bean, sesame, and the respective also oils and sunflower seeds, which are particularly rich in unsaturated fats. Nuts and seeds bridge the gap, providing concentrated forms of both energy and essential fatty acids. The type of source often influences the fatty acid profile, which is why dietary variety is very crucial for our diet. However, the question of sources isn't just about the nutrition, it's also about sustainability and global supply. Our need for new lipid sources is growing, driven by factors such as population growth, climate change, and food insecurity. The COVID-19 pandemic increased food prices, affecting especially developing countries. While enough food is produced globally, uneven distribution leaves close to 1 billion people hungry. Against this backdrop, oleaginous microorganisms such as yeast, bacteria, microalgae, and fungi are a promising source of sustainable lipid sources. This brings us naturally to what could be the role of biotechnology in producing these alternative lipid sources. In general, all microorganisms produce lipid compounds from structural and metabolic purposes, but only a few are able to accumulate them in amounts that can be considered commercially viable. Even within this same species, lipid yield can vary significantly depending on growth conditions like nutrient availability, temperature, and pH value. For microorganisms that are not oleaginous, excess carbon is typically channeled into other storage compounds like polysaccharides. So, which microorganisms have this ability to store substantial amount of lipid and how they are able to achieve this? Oleaginous microorganisms are defined by their ability to accumulate between 20 and 70% of their dry biomass as lipids under specific conditions such as nitrogen limitation. This process begins when cell division slows down, but the cell remains metabolically active, and the cells begin to convert carbon into fatty acid, triglycerols, and phospholipid. This lipid can be extracted after biomass harvest and used in a wide range of applications, making these microorganisms valuable biological factories. To better understand how they behave, it's useful to have a look on their growth kinetics under control conditions. So, as you can see in this growth kinetics in the slide, when nitrogen is abundant, we have the physiological growth, where oleaginous microorganisms prioritize cell division and biomass accumulation. When nitrogen becomes limited, a metabolic shift occurs, so instead of producing new cells, microorganism metabolism is turned into lipid accumulation. So you can see that, moreover, from this example, after 30 units, lipid accumulation starts to increase. And this pattern can be tracked over time in growth curves showing distinct phases of biomass growth and lipid accumulation. These kinetics are very important when we want to determine optimal harvest time in industrial proper production, meaning that we will have to see at which fermentation time we have to stop the process in order to have higher lipid accumulation, but also is important at this point to have also increased biomass production. We will see later on why this is important. Of course, lipid yield and composition are not determined only by the deficiency in nitrogen, but also there is a variety of other factors. Thus, several factors influence how much lipid can be produced by a microorganism. The genetic makeup of the organism sets the baseline, but also environmental conditions, for example, pH value, temperature, and light exposure, can shift both quantity and quality. Nutrient composition in the medium, especially the carbon and nitrogen sources, also plays a major role, as already discussed in the previous slide. Among eukaryotic microorganisms, only certain yeast and fungi have the genetic capacity to store high levels of triglycerols. For example, out of more than 600 known yeast species, only 20 or 30 are capable of accumulating tags in significant amounts. These lipids are stored in a very particular form inside the cell, something that we can actually see under the light microscope. So, the lipids can be synthesized de novo by oleaginous microorganisms, then stored intracellularly in the form of lipid droplets, which are clearly visible under a light microscope. Under nutrient depletion, these droplets can be mobilized and used as an energy and carbon source. The fatty acid profile of microbial oils are very similar to those of the plant-based sources, the vegetable oils, making them interesting alternative to oils produced through special crops. Now, let's continue with the main microbial groups capable of producing these lipids, starting from microalgae, such as Spirulina maxima, Chlorella, and others. These microalgae can accumulate up to 85% of their dry weight as lipids under specific conditions. They provide valuable polyunsaturated fatty acid, but also they are able to produce sterols and tocopherols with carbon chain lengths typically between 12 and 22 carbons. Environmental factors such as temperature strongly influence lipid content and also the saturation levels. However, their cultivation requires clean water and sufficient light. One commercial example is DHA gold, docosahexaenoic acid-rich oil derived from Schizohydrium microalgae. Another important group of lipid-producing microorganisms is bacteria, although their lipid profile is quite different. So, bacteria like Streptomyces, Bacillus, Acinetobacter, Nocardia produce complex lipids like glycolipids, phospholipids, acylglycerols, and only a few species are able to produce triglycerols. Their lipid profile are often complex and variable, and yields are generally lower than in microalgae or fungi. Additionally, some lipid-producing bacteria are considered as pathogenic and may produce toxins or allergenic compounds. This factor, combined with the lower lipid yields, limit their industrial use compared to the other microbial microorganisms that can be used as lipid-producing microorganisms. Now, from bacteria, we go on with the fungi, which can yield some of the most valuable polyunsaturated fatty acids. Certain fungi, including species from the genera of Mucor and Aspergillus, are capable of producing high polyunsaturated fatty acids such as DHA, EPA, linolenic acid, and arachidonic acid. Under optimized growth conditions, lipid content can reach up to 70% of their dry weight. However, cultivation have some challenges. For instance, the submerged culture can develop rheological problems, while the solid state fermentation requires precise control of the humidity. Scaling up is also demanding, especially when handling fungal mycelium. Nevertheless, the nutrition and industrial potential of fungal oils make them a valuable target for further development. The next slide presents the yeast, which offer an interesting combination of high yields, fast growing, and adaptability to diverse substrates. Oleaginous yeasts such as Candida, Cryptococcus, and Rhodotorula are able to accumulate between 25 and 70% of their dry biomass as lipids. They produce triglycerides, phospholipids, sterols, and in some cases, polyols, carotenoids, and organic acids. Their major fatty acids include palmitic, palmitoleic, stearic, oleic, and linolenic acid, with some small amount of myristic and linolenic. Yeasts have some, the use of yeast for lipid production have some advantages, such as the short life cycle. Also, they have minimum space and labor requirement, and can be used a wide range of carbon sources, from pure sugars to industrial bioproducts and waste. They are also highly amenable to genetic modification, enable tailoring fatty acids profile so as to have specific applications. Given these advantages, yeast can be considered true cell factories for lipid production. So, when we refer to oleaginous microorganisms as cell factories, we are emphasizing their engineered or optimized capacity to convert raw substrates such as sugar-based substrates into valuable lipid products. By controlling their environment, in some cases altering their genetic makeup, we can direct metabolic pathways toward the high yield of lipid production. This approaches integrates various fields such as microbiology, biochemistry, and process engineering in order to achieve both efficiency and sustainability in lipid production. So, as you can see in this slide, except from the de novo synthesis, which is triggered by nitrogen limitation and carbon excess, we have also the ex novo synthesis, at which as a substrate are given fatty acids, triglycerols, sterols, sterolyzers, and other lipid substrate, which at this phase the cell are able to absorb hydrophobic substrates, and the lipids oxidized for energy or stored as triglycerols. So in general, at the de novo synthesis, we have the production of lipids from the scratch, while at the ex novo, we upgrade and store the microorganisms upgrade and store external lipids. Now, at this slide, we can see how fatty acids and generally the lipid can be produced in microorganisms starting from the glucose. So the first step, at the first step of the glycolysis, glucose is broken down through this pathway in order to produce pyruvate, which enters the mitochondria. Afterwards, in the mitochondria, pyruvate is converted into a ketyl coenzyme A, which is the key building block for lipid synthesis. Because a ketyl coenzyme A is not able, due to its size, to cross the mitochondrial membrane, it is converted to citrate, which is a smaller molecule able to transport it to the cytoplasm and then reconverted to a ketyl coenzyme A by ATP citrate lyase. The lipid synthesis also requires NADPH for energy and reducing power. This comes from the pentose phosphate pathway, as you can see in this slide also, from enzyme such as malic ones. The final step is that a ketyl coenzyme A and NADPH feed the fatty acid synthase complex, which produce fatty acids that are assembled into lipids. Interestingly, in oleaginous microorganisms and under nitrogen limitation, AMP deaminase enzyme can be activated, breaking down AMP to provide ammonium as a nitrogen source. So, we have a reduction in AMP, which inhibits isotrate dehydrogenase, causing isocitrate and then citrate to accumulate. Citrate is exported to the cytoplasm, as said earlier, and then is further converted to a ketyl coenzyme 8, and lipid synthesis then is strongly enhanced, shifting the metabolism from biomass growth to lipid accumulation. So, in general, in oleaginous microorganisms, what differentiates them from non-oleaginous microorganisms is their ability to produce and continuously supply cells with ketyl coenzyme A molecules, and also they have the ability to produce sufficient amounts of NADPH for the biosynthesis of fatty acids. Afterwards, successive desaturation and chain elongation reactions occur, and the amount and type of fatty acid depends strongly on the genes present in the genome of each microorganism used for this purpose. Now, let's go on with the ex novo synthesis in the oleaginous microorganisms, which refers to the lipid production using hydrophobic substrates such as esters, triacylglycerols, fatty acids, and sterols. The process generally begins with the lipase enzyme secretion, which hydrolyzes this substrate into free fatty acids. This can be oxidized for energy or esterified into storage lipids. So we have two things, either oxidation for energy or used for storage after re-esterification. Some microorganisms can selectively metabolize specific fatty acid, enabling modification of the lipid profile during fermentation. This capability allows the design of oils with tailored composition when we want to use them for specific purposes in nutraceutical, pharmaceutical, or other uses. Of course, beyond the biology, we should also take into consideration the economic aspects of lipid production. Therefore, in bioprocessing, three key parameters determine economic viability. These are the selectivity, the yield, and the productivity. Selectivity refers to the ability of microbial cells to preferentially synthesize the desired product under specific conditions at the expense of other major intermediate or final products. Yield measures how much product is generated per unit of substrate, while productivity reflects the rate of this production over time. These parameters are very important because by optimizing this, producers can reduce cost and improve competitiveness, particularly when working with low cost or waste-derived feedstocks. Now we will examine more closely the specific factors that may influence the lipid production in oleaginous microorganisms. The carbon source is the most critical one. Common options include glucose, glycerol, xylose, sucrose, molasse, and also some hydrolysates of lignocellulosic material. The carbon to nitrogen ratio is also vital since high ratio lead to nitrogen depletion, which triggers the lipid accumulation. Additionally, oxygen availability influences fatty acid desaturation, while its strain has also optimum pH value and temperature ranges, which may yield to higher lipid production. In addition to this, there are also some micronutrient requirements and inhibitory effects that we should take also into consideration. Micronutrients such as magnesium, zinc, and iron can influence the enzyme activity during lipid biosynthesis. On the other hand, inhibitors, for example, furfular or phenolic compounds found in some complex substrates, may reduce yields by interfering with microbial metabolism. Successful process design involves balancing nutrient supplementation with strategies in order to remove or neutralize inhibitor. When all of these parameters are optimized, lipid yield can be maximized and fatty acid composition tailored for intended application. Now that we have seen some biological and chemical factors that influence the lipid accumulation, we will have a broader look at the industrial workflow. So the industrial production of microbial lipids, similar to the production of other microbial compounds, have three main stages. The upstream process, covering all the activities before fermentation, including the selection of the strain, the preparation of the substrate, such as the carbon source that we said, and also the development of the inoculum. The bioreactor stage is where the fermentation occurs under specific conditions. Then we have the downstream processing, which involves the recovery, the purification, and, if necessary, the modification of the lipid products. When optimizing each stage is very essential for overall efficiency and cost effectiveness. With this overview in mind, now let's break down the upstream and downstream processes. Shortly, the upstream process involves the preparation of the conditions that will allow the microorganisms to thrive and produce lipids in efficient amounts. This includes the selection of the strain, or, if needed, the genetic manipulation, the optimization of the growth medium, and ensuring that the correct environmental parameters, for example, temperature, pH value, and oxygenation is also very important, have been set from the start. Feedstock, especially waste-derived ones, may require some pre-treatment in order to remove inhibitors, such as phenols, as we referred to previous slides, so as to make nutrients more bioavailable for the microorganisms. A well-designed upstream process lays the foundation for high yield and consistent production. Consistent production is also very important in industrial processes. Once fermentation is completed, attention turns to the downstream process, at which we should recover and refine the lipid compounds for a specific use. So the downstream process begins after the end of the fermentation, at which the desired lipid compounds have been produced. Thus, the first step is the separation of the microbial biomass from the culture medium, typically by means of centrifugation or filtration. The next step, lipids can be extracted from the cells, often using mechanical disruption, and followed by solvent extraction or some more convenient environmental friendly methods, such as supercritical carbon dioxide. Finally, the crude lipid product may be purified, fractionated, or chemically modified, depending on the intended application. At this point, I have also to say that the efficiency of the downstream processing directly impacts both product quality and overall production costs. Now, let's look at where the single cell oil can be actually found application. First of all, in the food and feed industry, single cell oils are considered a valuable source of polyunsaturated fatty acids. These compounds are essential for example, infant development and functional food. Moreover, single cell oil offer a sustainable alternative to fish oils, to fish oils, free from marine contaminants, and they can be incorporated into nutraceuticals and other pharmaceuticals, functional foods, and animal feeds. In aquaculture, for instance, these oils improve the nutritional quality of fish without putting pressure on wild fish stocks. Moreover, we have the production of lipid for biofuels. So in the biofuel sector, single cell oil can be converted into biodiesel through the transesterification process, producing fuel with fatty acid profiles similar to those of vegetable ones. This offers a sustainable pathway for replacing fossil fuels, particularly in the feedstocks from waste-derived substrate. Additionally, single-cell oils may serve as a precursor for surfactants, lubricants, cosmetics, and bioplastics. In pharmaceuticals and cosmetics, high-value polyunsaturated fatty acid from single-cell oils can be used for cardiovascular health, anti-inflammatory treatments, and neurological support. Specialty oils are formulated into skincare products for anti-aging properties, and also moisturizing effect. Additionally, integrating this single-cell oil into biorefineries allows for co-production with other valuable products such as pigments, enzyme, and organic acids. Thus, through this integrated process, we can maximize economic and environmental benefits. At this point, it's good to mention that generally saturated and monounsaturated fatty acids are mainly produced and used for biodiesel production, while the polyunsaturated fatty acids are found application in nutraceuticals. In order to appreciate the scale of this opportunity, it's worth noting that the microbial lipid production is no longer just a laboratory exercise. It's already happening at industrial scale. So microbial lipid production has reached a commercial implementation, particularly for products rich in rare polyunsaturated fatty acids with high nutritional value. This includes both established oleaginous microorganisms and newly discovered strains. There is a growing scientific interest in these lipids due to their health benefits for humans and the other higher microorganisms. Industrial processes also often include the production of carotenoids, which are valuable bioactive compounds having both nutritional and commercial value, like beta-carotene and lycopene. However, alongside with these opportunities, there are some practical advantages and also challenges that need also to be mentioned in this lecture. So one major advantage of microbial lipid production is that it's not seasonal, and it is independent of climatic, ensuring a stable supply throughout the year. Bioprocess can also achieve higher yields and purities through metabolic regulation, and recovery costs are often lower than that of the conventional oils. Using low-cost raw materials such as the waste streams can further reduce the cost of the bioprocess. However, the main challenge is to achieve yields and process efficiency that meets industrial and cost competitiveness requirements, especially for bulk markets like biofuels. This naturally leads us to one of the most promising cost saving strategies, which is the use of food waste or agro-industrial bioproducts as a substrate. Since substrates account around 40 to 80% of the total cost of microbial lipid production, these food waste streams offer a low-cost, abundant, and alternative choice that also addresses environmental issues linked to the waste disposal. Uh, Globally, over 1.3 billion tons of food are wasted each year. So, food waste, either in liquid or solid form, which contains carbohydrates, proteins, and other nutrients, can be hydrolyzed into smaller forms that microorganisms can use. So, oleaginous microorganisms can convert this waste into single-cell oils, contributing to circular economy and bioeconomy. Beyond this concept, now let's have specific examples of food waste sources that can found such application. So some examples, as you can see in the present slide, include fruit and vegetable peels, pulp, expired juice, some dairy byproducts such as cheese whey, brewery wastewater, waste cooking oil is also very important. And by using this one, we can have also the ex novo synthesis, canning syrup and starch waste. The advantage is that these substrates are rich in carbon and other nutrients and can find application in fed pads or co-fermentation and solid-state systems. And another example is the use of waste cooking oil that can be used as a direct carbon source for Yarovia politica. And co-fermentation with other food waste may enhance the lipid yield and also reduce the waste disposal costs. So in order to make the most of food waste, we need to optimize the process from the beginning. Pretreatment, though, is often necessary in order to make food waste substrate more accessible to microorganisms. Various oleaginous microorganisms prefer simple sugars like glucose, so breaking down these complex carbohydrates, proteins, and fibers is a very crucial step. Without the pretreatment of the substrate, lipid yields can be very, very low. So enzymatic hydrolysis, acid treatment, or combinations like ultrasound and enzymatic hydrolysis or chemical and hydrothermal and enzymatic may increase the glucose release up to 4 times and also reduce the enzyme requirements. However, these methods can add cost and in some cases generate secondary pollutants. So process design must balance efficiency and sustainability. Pretreatment is just one thing that we should take into consideration. Other process optimization strategies for lipid production from food waste is nitrogen limitation, which is already said, triggers the lipid accumulation, redirecting metabolism from biomass production toward storage lipid synthesis. Combining different substrates, for example, glucose with waste cooking oil, we may improve the carbon balance and increase total lipid accumulation. From the genetic point of view, engineering strains like Yarovia politica may also enhance lipid biosynthesis and also reduce sensitivity to inhibitory compounds found in waste streams. This integration of biochemical process and genetic optimization may significantly improve the economic viability of microbial lipid production from food waste. Finally, to close this section, this lecture, we will consider some of the challenges and future direction for using food waste as a feedstock. So, despite its potential, using food waste for microbial lipid production, we have also some challenges. The composition is highly variable depending on season, source, and requires constant pretreatment and process control. This complex structure means that it's not directly usable by homogeneous microorganisms. So carbohydrates must be broken down to sugars, protein, and amino acids. Moreover, some waste contain inhibitors such as peroxides or aldehydes that also, like phenols mentioned before, may hinder the metabolism of the microorganism. So post-processing may also be an issue as solid residues require disposal and purification cost can be high in order to achieve industrial-grade lipids. Looking ahead, research is focusing on ways to overcome these barriers and make the process more robust. So future strategies may include the strain engineering in order to tolerate and efficiently metabolize complex or inhibitory waste streams. Synthetic biology tools can also adapt microorganisms like Yerovia lipolytica for hydrophobic waste use. Process optimization will aim to improve transformation efficiency and scalability, while techno-economic assessments will help determine cost-benefit feasibility for industrial adoptions. Finally, integrating lipid production into larger biorefinery systems may maximize resource use, contributing to a circular bioeconomy where multiple products are generated from the same waste feedstock. Now, we will end up with 3 examples of lipid production from food and agro-industrial wastes. The first one is olive meal wastewater, which is a bioproduct of olive oil production, rich in organic matter and also phenolic compounds. It contains sugars, organic acid, lipid, and polyphenols. The yeast, for example, Cryptococcus curvatus, is able to grow on pre-treated olive meal waste water and may lead to high lipid yield up to 45% of their dry weight. One challenge is the presence of phenolic compounds, which may hinder the microbial growth, though pre-treatment often is required. Using olive meal wastewater as a feedstock not only produces valuable single-cell oil, but also help reduce the environmental pollution associated with its disposal. Another widely available waste from the food industry is cheese whey, which has its own characteristics as a substrate. Cheese whey is generated in large quantities during dairy processing and contains high level of lactose, proteins, and minerals. Most oleaginous yeast cannot directly use lactose, so hydrolysis is required in order to convert it into fermentable sugars. Once this is done, substrate like whey hydrolysate can support the high lipid accumulation, with fatty acid profiles similar to those of vegetables ones. This makes it a promising source for biodiesel feedstock and value-added nutritional lipids, while also addresses the disposal problem faced by this dairy industry. Finally, let's revisit food waste, which is in a more general sense, looking at its composition, microbial use and benefits. Food waste streams, whether from households, restaurants, or industrial food processing, are rich also in carbohydrates, proteins, lipids, vitamins, and minerals. Pre-treatment of this waste releases nutrients, making them accessible to lipid producing microorganisms. For example, Rhodosporium tourlodes grow on enzymatically pretreatment food waste can achieve lipid contents up to 60% of their dry biomass. The main challenge is the heterogeneous composition, which requires its time, adaptation to microbial strains, and also consistent processing. Nevertheless, using food waste from lipid production supports the circular economy by turning discarded resources into viable bio-based products. Now, before the end of this lecture, I would like to kindly ask you to take a few minutes in order to evaluate our lecture, which is very important for the AGRIMA project. So please scan the QR code or type the link that you see in this slide for the evaluation. I would like to thank you for your attention, and I hope that you enjoyed this presentation. Thank you.